some of the data on KO-539 was presented at last year's American Society of Hematology meeting. KO-539 is a selective inhibitor of menin, which interacts with a key proteasome enzyme called KMT2A. And in this pathway, the interaction of menin with KMT2A appears to select for aberrant suppression of myeloid differentiation and maturation, leading to the phenotype of AML. So KO-539 and other menin inhibitors like it interfere with the interaction of KMT2A and menin, leading to the release of the block in differentiation and ultimately maturation and differentiation of myeloid malignant cells, and hopefully a therapeutic impact in leukemia. Most of the data that has emerged with menin inhibitors from the compound from a company called Syndax and this one from a company called Cura have been early and preliminary. So there's only been a dozen patients, for example, in the KO-539 study. Among the dozen patients, we reported 2 patients with very robust complete remissions, one of which was MRD negative, so measurable residual disease negative. That patient that happens to be at my institution continues to be in remission, so continues to do well despite having had 7 prior regimens for relapsed refractory disease. So it's quite remarkable. It appears that this drug would be expected to be more active in patients who have NPM1 mutations or KMT2A or / also known as MLL rearrangements. But one of the patients who achieved remission on the study to date did not have those alterations, but did have a SETD2 mutation, which seems to play a role in that same complex of MLL and menin that I briefly alluded to earlier. The drug KO-539 appears to be quite well tolerated without any clear and established trend in toxicity, but we will have to see if this efficacy trend continues in a larger cohort of patients as the study progresses.